Hey, welcome to The Explainer. Today we're doing something pretty cool. We're going to shrink ourselves down, way, way down, to the microscopic level. Why? To check out one of the most brilliant and honestly essential structures in all of biology. We're talking about the boundary that literally separates life from non-life. Let's dive in.
So let's start with a big question. What actually separates a living cell from, well, the non-living world all around it? I mean, really think about that. Every single living cell, whether it's a tiny bacterium or one of the neurons firing in your brain right now, has to keep its inside world just right, completely separate from the chaos on the outside. So what is it? What's the gatekeeper, the bouncer at the club door? What is this intelligent border that makes life itself possible?
Well, the answer is something called the biological membrane. And let me tell you, this is not just some simple wall. It's a dynamic, it's a fluid, and it is an incredibly smart barrier. This membrane is the whole reason a cell has an identity. It protects it, for sure, but it also manages this constant, bustling flow of traffic. Stuff going in, stuff going out.
Okay, so to really get how the membrane pulls off this incredible job, we first have to look at what it's made of, right? Because its structure, that's the secret to everything it does. So let's peel back the layers. So you've basically got two main players on the team here, lipids and proteins. The lipids, they form this flexible, continuous double layer. We call it a bilayer. Think of that as the basic container, the fabric of the membrane. But studded all throughout this oily fabric, you have the proteins. And these are the real game changers. If the lipids are the fence, the proteins are the intelligent gates, the sensors and the channels that decide who gets in and who stays out. This brings us to this awesome concept called the fluid mosaic model. And it's a perfect name because the membrane isn't some rigid, static wall. Nope. It's more like a bustling sea. It's a dynamic, two-dimensional fluid. The lipids are constantly shifting and swapping places, and those proteins? They're like big icebergs just floating and drifting around in this lipid sea. I mean, scientists have even done experiments where they fuse two cells together, and you can literally watch the proteins from each cell just mix and spread out across the new surface. It's incredible. And the genius of this whole design, it really just comes down to basic chemistry. See, each of these little lipid molecules has a water-loving head and a water-hating tail. So what do? They automatically, spontaneously arrange themselves into that bilayer with all the water-heating tails tucked away on the inside, creating this oily core. And that core is what makes it such a fantastic barrier, because it naturally repels almost anything that dissolves in water, like salts and sugars. It just works. All right, so if this membrane is such a great barrier, how in the world does a cell get all the good stuff it needs in and get all the waste out? Well, it all comes down to the rules of entry. And it turns out there are two fundamentally different ways to cross this border. Okay, check this out. On one side, you've got passive transport. The easiest way to think about this is like rolling downhill. It takes zero energy. Molecules just naturally move from where there's a lot of them to where there's not so much. Simple. But then you have active transport. This is the complete opposite. This is like pushing a boulder uphill. It takes a ton of energy, and it lets the cell force molecules to go against their natural flow, from a low concentration to a high one. So who gets to just waltz right in, and who needs a special passport? Well, small, uncharged molecules like oxygen and CO2, they're tiny enough to just slip right through the lipid bilayer. No problem. That's passive diffusion. But for pretty much everything else, we're talking water, glucose, and especially charged ions like sodium or potassium, that oily core of the membrane is a total roadblock. They absolutely cannot get through on their own. They need that passport. They need an escort. And that comes in the form of those protein channels and transporters we were talking about. Okay, now this is where it gets really cool. A cell doesn't just want to be in balance with the world outside. No way. It needs to create and maintain these really specific high concentrations of certain things inside. And to do that, it has to actively fight against that natural downhill flow of diffusion. It has to pump things uphill. And this is such a crucial point. To create order and function, a cell has to actively pump things against their natural tendency. It has to fight. Creating that special internal world that makes you, you, well, it's a constant battle against chaos. And that battle costs energy. It is a fundamental cost of being alive. So where does all this energy come from? It comes from the cell's universal power source, a molecule called ATP. The best way to think of ATP is like a tiny, fully charged battery.
Whenever the cell needs to do some serious work, like pumping a molecule uphill against its gradient, it just spends an ATP molecule, breaking it apart to release a perfect little packet of energy to get the job done.
And probably the most famous, most vital example of this is the sodium-potassium pump. This protein is an absolute workhorse. It never stops. For every single molecule of ATP it burns, it tirelessly pumps 3 positively charged sodium ions out of the cell and then grabs 2 positively charged potassium ions and pulls them in. Now, this might sound like a small thing, but get this. This one type of pump can be responsible for up to 1/3 of all the calories you burn just sitting still, all just to maintain this crucial imbalance.
So why do all this work? What's the point? Well, think about it. You're kicking out 3 positive charges for every 2 you bring in, over and over. That makes the inside of the cell just a little bit negative compared to the outside. This electrical difference, this voltage across the membrane, is called the membrane potential. And it is not just some random side effect. It is the entire point. This stored electrical energy is the foundation for one of the most incredible things in all of biology, how your neurons fire a signal. So, a neuron that's just hanging out, waiting, has a membrane potential of about -70 millivolts. It's just sitting there, polarized like a tiny battery that's fully charged with potential energy, just waiting for the signal to go.
And here comes the action. When a stimulus finally arrives, it could be a touch, a sound, a chemical signal from another neuron. It triggers these special protein channels to fly open. These are the voltage-gated sodium channels. They're normally locked down tight, but that stimulus is the key.
And with those gates thrown wide open, all that sodium that the cell has been pumping out rushes back in, just flooding the cell, following its natural gradient. This torrent of positive charge completely flips the script. The membrane potential skyrockets from -70 millivolts all the way up to +50, and it happens in a fraction of a second. This massive, dramatic electrical spike, that is the action potential. That's the nerve impulse. It is the signal. And it doesn't just stop there. This sudden flip in voltage triggers the next set of sodium channels a little further down the axon to snap open, which triggers the next set, and the next. It creates this incredible chain reaction, this electrical wave that just zips down the length of the neuron at unbelievable speed. That's how a thought can feel instantaneous. But what happens when that electrical wave gets to the very end of the neuron? Well, this is where it gets converted into a chemical signal. First, the action potential arrives at the axon terminal. That arrival triggers new gates to open, this time for calcium. Calcium rushes in, and that acts as the signal for these tiny little bubbles, they're called vesicles, that are jam-packed with chemical messengers. The calcium tells them to fuse with the outer membrane, and poof! They release all those neurotransmitters into the tiny gap, carrying the signal over to the next cell. And that really brings us full circle, from the simple chemistry of a lipid bilayer all the way to the firing of a neuron that creates a thought. It all just comes down to the controlled movement of ions across a membrane. I mean, every single thought, every feeling, every action you take is literally powered by this process. So if that's true, what does that make us? It's a pretty staggering thought and an amazing testament to the power of the cell's dynamic border.